Very good morning. A warm welcoming to all the dignitaries, faculty members, Honorable Principal of SRR Government Arts and Science College Autonomous, Professor Kalvakunta Ramakrishna Garu, and another legendary personality, Dr. Jaikishan Oja sir, Principal, GDC Godavari Kani and Mantani. Today's keynote speaker, eminent personality, eminent Professor Dr. Dhale sir, and other organizing committee members, respected Vice Principal sir, Dr. P. Nitin sir Garu, HOD Physics Department, and Dr. Talapalli Rajaya sir, Head Department of Head Department of Commerce and Business Administration and Board of Studies Chairperson and Vice Principal of SRR Government Arts and Science College. And today's webinar organizing secretary, Dr. Rautu Radhakrishna sir Garu, and co-conveners of this webinar, Dr. P. Savita Madam Garu, GDC Godavari Kani, Dr. Muhammad Tahir Hussain, Dr. Kandukuri Kalajyoti Madam Garu, Dr. Asampalli Swaruparani Madam Garu, Dr. Muhammad Tajuddin, and Dr. Ramya Madam Garu, and other faculty members of GDC Godavari Kani, GDC Manthani, and our esteemed college, SRR Government Arts and Science College. And one of the most important personality for organizing this webinar, that is none of the other than person, Dr. Thirukovela Srinivas Garu, Head Department of Botany, Microbiology, Head Department of Botany, Food Science, Crop Production, and Controller of Examinations of SRR Government Arts and Science College, Autonomous, and all the dignitaries from various reputed institutions from our Telangana state, and also the other various universities from our sacred nation. Wholeheartedly welcoming you. This is Dr. PADALA Tirupati, Assistant Professor, Department of Botany, Food Science, Crop Production, and co-organizing secretary of this webinar. So wholeheartedly welcoming you. Before start our webinar, I will going to show some of the glimpses of our botany department, related to our botany department. I hope this slide is visible. The screen is visible. Okay. So this is our slides. This is our webinar flyer. Sorry, I forgot. Today's our chief guest. Today's our program chief guest, Dr. Tirunahari Yugender sir Garu, Associate Professor, Department of Botany, Government College, Kakatiya Government College, Hanumakonda. Wholeheartedly welcoming you, sir. Slide show కనబడుతుందా sir? Okay. So this is one of the program is organized in our department, especially microbiology department, in the presence of respected principals and also the organizing secretary of today's program, that is Dr. Rautu Radhakrishna sir, microbiology, head department of microbiology. So this is one of the functions celebrated in microbiology department. It is an offshoot of the botany. This microbiology head post as charge has taken by our Department of Botany, eminent personality Dr. Rautu Radhakrishna sir. So microbiology department recently organized the best program, one of the workshop, that is the Microscope Odisha program has conducted with experimentally, and they are showing in innovative way our honorable principal and other faculty members are appreciated the program. And Batkama celebrations also celebrated in the Department of Microbiology. So this slide shows about the Telangana Floral Festival. The entire world floral festival has conducted or celebrated in our Telangana state only. So this slide shows about our proud of Telangana, that is Batkama Sambaralu, the floral festival. So recently, Dr. Tirunahari Yugandhar sir, Associate Professor, visited our Department of Botany on 5th September, the Teachers' Day, on Teachers' Day. So our head of the Department of Botany as felicitating the eminent personality, Dr. Tirunahari Yugandhar sir. And in our Botany Department, as sent the Jignasa Student Study Project in the presence of the jury member, that is Dr. Nalla Manoj sir, Head Department of Botany, GDC Women Autonomous Karimnagar. On that day, our students are presented Jignasa Student Study project. Dr. Rajesh who worked in GDC Godavarkan in earlier as a FAC. So sir is also now the coordinator of student Jignasa study project of our SRR Government Arts and Science College. On one fine day, recently, Patriji, Subhash Patriji poster has released in the Department of Botany in the presence of our head Department of Botany. And this slide shows about the campaigning program for admission into 2025 and 26 for our reputed institution. Our faculty member, Dr. Asampalli Swaruparani Madam Garu, is explaining the facilities and also the courses regarding of the new students, those who are going to attend intermediate public examination second year. So this is the pamphlets they are distributed among the students of Jamikunta Junior Colleges. And recently we are organized one national webinar on intellectual property rights for academics and research in higher education. Professor Togapur Pavan Kumar sir enlightened on that day. So this slide shows about how the concern about the Department of Botany. Recently, our head Department of Botany, Dr. Tirukovela Srinivas Garu, purchased one pipe for the protecting the new saplings planted in front of the Botany Department. So we assured very soon our Botany Department in front of the Botany Department and also the Dr. B.R. Ambedkar seminar hall in front of the seminar hall. Definitely with the support of our honorable principal, Professor Kalvakunta Ramakrishna sir, I am assured this entire area is going to green very soon, will show our responsibility. Okay. Vachinda? Right. So recently in the Department of Botany, we are celebrated the Food Fiesta Festival, Food Fiesta for Healthy Diets. By the suggestion of our head Department of Botany, Dr. Tirukovela Srinivas Garu, our PG students are prepared different dishes along with different type of the fruits, rainbow colors of the fruits for our healthy life. From after COVID-19, we are cautious about our health. So that's why our Department of Botany and even though food science, our Department of Botany is also responsible for the healthy diet awareness programs are conducted among our 800 botany students, and also by the concern of support of our head Department of Botany, in his car, our students are the entire dishes and the fruits are carrying through car and established one stall in the Fairy Fiesta celebration has conducted in the GDCW Autonomous Karimnagar. Our students are established one stall and by the cooperation of Asampalli Swaruparani Madam Garu and Kandukuri Kalajyoti Madam Garu, who supported for the students, and Honorable Collector Madam Garu, Srimati Pamela Satpati Garu, and the Varalakshmi Madam Garu, Honorable Principal of GDCW Karimnagar, appreciated for their commitment. And the this is the press clip as covered our food festival is conducted in the Department of Botany. And recently, our Department of Botany, Crop Production, Food Science and Quality Control, along with the Zoology Department and Fisheries, we are conducted a campaigning program for 2025 and 26 new academic year, visited very nearer in the locality of the Karimnagar Junior Colleges, that is Gaud Junior College for girls and Science Wing, Government Junior College Science Wing, and Arts and Commerce College in Karimnagar. We are given the distribution of the pamphlets among the people. Our Kandukuri Kalajyoti Madam Gar has distributed these pamphlets among the students. So very recent, before day 31st January, our senior assistant, very kind-hearted personality, Shankarama, as retirement slide showing in this slide. Our honorable principals are addressing the program. And yesterday, we are participated 1 month road safety awareness program closing ceremony from our compound, from our the ground, SRR College ground. Our honorable principal, Professor Kalvakunta Ramakrishna sir, and along with our honorable district collector, Srimati Pamela Satpati Garu, has inaugurated this program, and our NSS volunteers and faculty members and program officers, they are participated rigorously. More than 800 bike rally from our ground to near Gita Bhavan, and the importance of the helmet, wearing helmet and road safety information has sent in among the people. And very recently, yesterday, the Deputy Commissioner of Transport, Road and Transport, Vamshidhar Garu, and the other officers are visited our Department of Botany. Regarding of that time, our Honorable Head Department of Botany, Dr. Tirukovela Srinivas sir, has felicitated the eminent personalities who visited yesterday in our botany department. So the blue t-shirt, we are observed very young and dynamic personalities, none of the other than our honorable principal, Professor Kalvakunta Ramakrishna Garu, along with the collector program yesterday in bike rally participated, and the collector madam has appreciated entire faculty members and NSS program officers and Department of Botany yesterday program. See how the concern and the who involved in the bike rally program, young and dynamic personality with blue t-shirt, young person looking the told about the how impact of the helmet in our life during journey. So without helmet, we don't want to move any other place. Sir has showed and ride a bike yesterday. So this is the one of the press clip yesterday's covered news I presented here, and with this signature of our honorable principal, we are going to conduct this one-day national webinar. So without this signature, we are unable to conduct this program. So that's why we are wholeheartedly thankful to our honorable principal, Professor Kalvakunta Ramakrishna Garu. Thank you very much. So now the celebrating time of the botany department, SRR food court here, and our head department, we are wholeheartedly thankful to head department of botany, Dr. Tirukovela Srinivas Garu. These are the botany faculty members celebrating Uttappa Day. So thank you very much. These are some of the glimpses of our botany department. So now I would like to invite Professor Kalvakunta Ramakrishna Garu. Before message about our chief patron of this one-day national webinar, I will I will explain about the two legendary principals, and they are the coincident that both of they are organizing or mentoring and directing two reputed institutions. So Professor Kalvakunta Ramakrishna Garu, principal of our SRR Government Arts and Science College, along with the another autonomous college, that is GDCW Jagityal Autonomous College. ఒక కాలేజీని అటానమస్ కాలేజ్ చేసి మరొక అటానమస్ కాలేజీకి వెళ్ళినటువంటి రెండు కళాశాలలను లీడ్ చేస్తున్నటువంటి మన ప్రిన్సిపల్ డైనమిక్ పర్సనాలిటీ ప్రొఫెసర్ కల్వకుంట రామకృష్ణ గారు. Along with this legendary personality, the another legendary personality, legendary personality, principal, Dr. Jaikishan Oja Garu, who took the charge as a principal of GDC Godavari Kani, along with GDC Mantani. So that is two. Because of this is that second national webinar, our two principals took the charge, two colleges. So that is the coincidence of this second national webinar. So we are very lucky and we are very happy to invite today's chief patron of this one-day national webinar, second national webinar, Professor Kalvakunta Ramakrishna Garu. Please welcome and unmute yourself and give a brief message regarding of this one-day national webinar. Please welcome, sir.
Thank you, Tirupati. Am I audible?
Yes, sir. Yes, sir. You are audible, sir. Please proceed.
I warmly welcome to one-day national webinar on pharmacognostic studies of medicinal plants. Department of Botany, SRR Government Arts and Science College, in collaboration with GDC, Godavari Khani. It is a great honor, enthusiasm. Department of Botany recently organized one national webinar with association GDC Kamareddy. Now, association with GDC Godavari, my beloved friend Dr. Jaikishan Oja, principal of GDC Godavari and Mantani, and all organizers and HODs of both colleges, Dr. T. Srinivas Garu and GDC Godavari Botany Department faculty members, SRR faculty members, Tirupati Garu and Srinivas Garu, Radha Krishna Garu, Kala Jyoti Garu, Dhale Garu, another Swarupa Garu, all my beloved colleagues. I warmly welcome, extend my warmest greetings to all the participants. One-day national webinar on this topic, pharmacognostic studies of medicinal plants. We know the significance of the topic. This platform is very useful for the worldwide of medicinal plants and their pharmacognostic potentiality. Medicinal plants have played a vital role, crucial role in healthcare system across the globe for the centuries. The contribution to traditional medicine and modern drug discovery are invaluable. The field of pharmacology allows us to understand their therapeutic properties, chemical composition, a sustainable use for future generations. This webinar provides a remarkable opportunity for young research scholars and academicians and students to engage in insightful discussions, share their knowledge, and gain the deeper understanding of the scientific advancement in the medicinal plants field. We are honored to have Prof.Datha Dhale, keynote speaker. Sir, I warmly welcome. We have a principal and all the staff members and Department of Botany, SRR Government College, whose extensive knowledge and research in the field of pharmacology and undoubtedly provide immense value to all attendees today. His expertise will illuminate various aspects of medicinal plants studies, offering key insights into their scientific applications. My sincere appreciation goes to our beloved friend, co-partner, Dr. Jaykishan Oja, Principal Government Degree College, Godavarkani and Manthani, for his continuous support and guidance for the faculty members. His commitment to academic excellence and encouragement towards research programs initiatives instrumental to organize this webinar. I also very happy to privileged to have Dr. Yugandhar, my beloved friend. Previously, I worked with Yugandhar sir also. Today's main speaker and chief guest also. His contributions to botany and medicinal plants research are commendable, appreciable also. And these insights will inspire the participants to explore new dimensions and new innovative things in this viral area of study. A special note of gratitude to Dr. P. Srinivas, convener of this webinar, along with co-conveners and organizing secretaries, all the committee members, for their meticulous planning and tireless efforts in bringing this national level webinar. This webinar and their commitment to promoting academic excellence and research, scientific discussions is truly highly commendable. As we embark on this intellectual journey, I encourage all the participants to actively engage in these discussions, observe valuable knowledge and contribute their thoughts and perspectives. This webinar is a testament to our commitment and academic excellence and the advancement of pharmacognostic and studies. I extend my heartfelt gratitude to all the distinguished speakers and guests and participants of research scholars and students of both colleges being a part of this wonderful event. I am confident and assured that this webinar will be enriching experience of everyone involved, fostering new collaborations and opening avenues for future research medicinal plant sciences. I wish you all the best and an insightful webinar. Both the college organizing secretaries, highly appreciable. Thank you, my beloved friend, Dr. Jaykishan Oja. And I once again thank you for inspiring speaker, Professor Datta Dhale, a keynote speaker. Thank you, sir. Thank you, Vandal. Goes to Dr. Tirupati. Yes.
Thank you very much, sir. With these few words or then motivational words is enough to organizing these progressive activities in our college in the Department of Botany. Thank you very much, sir. Now, I would like to invite the another legendary principal of Government Degree and PG College Godavarikani and Government Degree and PG College Manthani, who established the new course previously in Government Degree and PG College Lakshadpet, and this is GDC Lakshadpet is one of the one and only model college in our the northern Telangana, and also in government institutions only 4 years integrated degree has established and successfully running that course, that is integrated teacher education program. That is the new program in our entire Telangana. So only government institution, that is GDC Lakshadpet, even though the ITEP program is the flagship program. Today news we are observed in the newspaper, only integrated teacher education programs are going to organize in future endeavor given by National Council for Teacher Education. So the visionary personality has established new course and convinced of the higher authorities from the NCTE and also the Mysore regional institute in Mysore. So they are satisfied to establish this new course, integrated teacher education program in GDC Lakshatpet, who worked for the progressive manner and also rigorously involved the all type of programs, including the not only the principal, but also is one of the voluntarily worked in the field of establishment of the new saplings and new plants, medicinal plants in the GDC Lakshatpet. Same in the same tempo has continue in the I observed whenever visited the GDC Godavari Kani as a flying squad team. Sir has also continued that type of new plants are established in the GDC Godavari Kani. So that is the inspiration and role model for the young faculty members of in higher education in the CCE Telangana. So with these few words, I'm very thankful to sir because of sir has selected as a coordinator of first coordinator of integrated teacher education program in GDC Lakshatpet. So that's why I'm wholeheartedly thankful to you forever for choosing me as a first integrated teacher education program coordinator. So I'm very thankful to you, sir. Now I would like to invite Dr. Jaikishan Oja, sir, and please unmute, sir, and give a brief message regarding of this one-day national program. Our association is always eternal. Thank you very much, sir. Please unmute.
Yeah, thank you, Tirupati sir, for your appreciations. Respected Ramakrishna Garu, Principal, Government Degree College, Women's Jagityal, as well as Principal for SRR Degree College. So I am very proud and feel privileged to be associated with this national webinar. Respected conveners of this national webinar, T. Srinivas Garu, Radhakrishna Garu, HOD of Microbiology and Organizing Secretary of this webinar, Tahir sir, again co-convener for this webinar, Kala Jyoti Garu, and Dr. Yugender Garu, Chief Guest for this webinar from KDC Warangal, other organizing members, and my special thanks to PADALA Tirupati sir. He had been associated with our college previously also and made us a part of many national webinars. So I thank to all the organizing members for this program. My special thanks to our keynote speaker, Dhale Garu from Dr. Ghogre Science College, Dhule, Jalgaon, and all the participants and faculty members from either GDC Mantini or Godavari Kanni, SRR and other colleges. I welcome you to all to this very important webinar. As you were saying, I love nature, I love plant. And again, sometimes I feel that I would be better as a botanist than a physicist. So I feel very happy to be associated with the webinar, Pharmacognostic Studies of Medicinal Plants. We know about pharmacognostic studies. It involves the investigation regarding the physical morphology of plants, yes, phytochemical analysis, yes, all this we do to study the plants which can be used as drugs. Nowadays, wherever we see, we speak about natural products. Yeah, we prefer natural products. We prefer herbal products. Yeah, so if you want herbal or if you want natural products, we have to study. We have to carry the study on the plants. So this webinar will be useful to the faculties of, I think, all the fields. And again, we are very happy to have a very eminent speaker for this webinar. I have gone through the research papers of Dr. Prof.Datha Dhale Garu. So he had published a number of papers right from 2011. It's continuous publications with good citations. Yes, I have seen the phytochemical screening of many of the species. Very useful plants. Yes, and again with good journals, he is having the publications. So definitely he is a very inspiring personality. So whoever have participated in the webinar, they should be lucky. Why? Because we'll be coming out with good knowledge and a good motivation. Okay, what we can do in future and how we can use this study, yes, to generate new drugs. And again, I feel really I feel privileged to be a part of this webinar. Why? Because we have a very dynamic leader as a patron for this webinar. Dr. Ramakrishna sir, he is a very leading personality in the region. Yes, he is a all-rounder. Throughout the day, he keeps busy from with academic as well as the social activities. So wherever we see, we find Ramakrishna sir. So definitely, he is a very inspiration for all the young principals or faculty members. So with this remarks, I thank all the organizers and all the participants, and I wish all the best. Thank you, Tirupati sir.
So really, this second national webinar is 100% fruitful. Why? Because so eminent legendary principals are here, and eminent professor, keynote speaker, Professor Dhale sir also associated with us, and the dynamic HOD, Department of Botany, Dr. Tirukovela Srinivas sir, associate professor, head Department of Botany, and also the major responsibility, that is controller of examinations of SRR Government Arts and Science College. Recently, sir has successfully released the complete terms of our SRR College results in the presence of our honorable principal, sir, Dr. Professor Kalvakunta Ramakrishna, sir. So with these words, Dr. Jai Kishan Vaja, sir, thank you very much. And now I would like to invite Dr. Thalapalli Rajaya Garu, Vice Principal of our college and HOD Commerce and Business Administration. So please welcome, sir, and unmute yourself. Dr. Rajaya, sir. Rajaya, sir. Hey, Jane. Leader. Dr. Rajaya, sir. Okay. Now I would like to invite the another vice principal of our college, Dr. P. Nitin Garu, head department of physics and vice principal of our college. Please welcome, sir. Okay. So now I would like to invite today's convener of this second national webinar, Dr. Tirukovela Srinivas sir, HOD Botany, Food Science and Crop Production, and Board of Studies Chairperson, and also the controller of SRR Government Arts and Science College. So please welcome, sir, and please unmute yourself and give a brief message regarding of this second national webinar. Please unmute yourself, sir.
Yes. Thank you, Tirupati. May I audible?
Yes, sir.
Yes. Good morning to all. It is with great pleasure that I extend a warm welcome to all participants, esteemed guests and scholars to this one-day national webinar on pharmacognostic studies of medicinal plants 2025. And this event has been thoughtfully organized by the Department of Botany, Crop Production and Food Science, SRR Government Arts and Science College Autonomous Karimnagar, Telangana, in association with Department of Botany, Government Degree College, Godavarikani and Manthani, Peddapalli District, Telangana, with the aim of bringing together researchers, academicians and students to explore the significance of medicinal plants and their pharmacognostic properties. Yes, we know that our patron sir, Oja sir, said that medicinal plants have been a crucial part of traditional and modern medicine, serving as an essential resource for a new drug discoveries and healthcare innovations. The field of pharmacognosy plays a key or a vital role in the understanding the physical, chemical, and biological properties of these plants. Certainly, this webinar aims to enhance our knowledge of medicinal plants, their scientific importance, and their role in the sustainable healthcare solutions. Here we are honored to have Professor Dhale sir as our keynote speaker. He is from Maharashtra. So he is a very kind and always motivational person. I personally met at Dhule, Maharashtra, and he is distinguished scholar and a researcher in the field of pharmacognostic. And his insights and expertise will surely provide valuable knowledge to all today participants. And our gratitude extends to our patron, Professor K. Ramakrishna, our beloved principal, and SRR Government Arts and Science College, Autonomous Karimnagar, and other co-patron, Sir Dr. Jaikishan Oja, sir, the principal of Government Degree College, Godavarkani, and Manthani, and our principal, vice principals, Nitin sir and Rajesh sir, their unwavering support and encouragement in organizing this webinar. We are also privileged to have Dr. Yugandhar as our chief guest. He is very a friend of all our faculty members, botany faculty members, and whose presence adds immense value to this event, and his contributions to the field of botany, especially biotechnology and medicinal plants research, are commendable, and we look forward to his valuable insights. And I would like to express my sincere appreciation to my fellow co-conveners, Padala Tirupati and his team, and dedicated organizing secretary Radha Krishna and his committee members for their hard working and commitment in making this webinar a success. Their efforts have been instrumental in planning and executing this academic event. Lastly, I extend my heartfelt gratitude to all the participants, research scholars, and students who have joined us for this knowledge-enriching session. I am confident that this webinar will provide a platform for meaningful discussions, knowledge sharing, and new perspectives in the field of pharmacognosy. I am wishing you all a fruitful and engaging session. Thank you, Tirupati.
Thank you very much, sir. So, with your support and encouragement, definitely our botany department shining like a star forever, and also the in our college and campus also going to green. So thank you very much, sir. But with your concern about our plans and department, definitely we're going to prove ourselves. So thank you very much, sir. So we expect from you so this type of encouragement and also the support for organizing this type of programs and also the seminars, workshops, symposia, and one more important information I will going to share by the suggestion of our honorable dynamic HOD Garu. Our department will going to celebrate the organizing the 2 days state level plant exhibition, that is very rare medicinal and herbal plants is going to organizing in our college campus. By one of the most eminent personality, Mokkala Venkataya from Kottagudem, Khammam district. Very soon dates will going to finalize by our HOD, Dr. Tirukovela Srinivas Garu. Definitely will going to organize this 2-day state level exhibition, plants exhibition, medicinal and herbal plants exhibition in the college campus. So thank you very much, sir. Definitely will going to organize. So thank you very much, sir. Now I would like to invite the Vice Principal of our college, HOD Commerce and Business Administration, Board of Studies Chairperson, Dr. Thalapalli Rajesh sir Garu. Please unmute yourself and give a brief message. Please welcome, sir.
Sir, good morning to all. First of all, congratulations to Department of Botany, Food Science, Crop Production, Microbiology of this SRR College, and Godavarkani College Principal Oja sir, and Botany Department, whole department. Congratulations, sir, for conducting this type of webinars to create awareness and to enlighten the importance of medicinal plants on the topic is Pharmacognostic Studies of Medicinal Plants 2025. And respected principal of our college, Dr. K. Professor Ramakrishna Garu, and today's keynote speakers, Datha Dhale, Professor Datha Dhale, and Dr. Gokre. So this topic is very interesting, and also it is not a new topic because medicinal plants, we know from the ancient area, our our ancient people are using, though they don't know what is the benefit of through the each medicinal plant. And in ancient days, Atharvana Veda also revealed that the regarding medicinal plants, and we know in ancient days, Dhanvantari, Sushruta, they are also using and suggested and revealed the importance of the medicinal plants. This is the time to increase awareness and to enlighten the medicinal plants, whatever Ayurveda, natural and herbal plants, whatever. One thing, sir, just I want to say, yeah, we are taking food for getting vitamins, nutrition, and on minerals also. But if whenever we are feeling unwell, we are going to consult a allopathy doctor. Why don't we get that medicine from these plants? అంటే మనకు ఉన్నటువంటి నమ్మకం అనేది ఈ మెడిసినల్ ప్లాంట్ల మీద అంతగా లేదు. ఎందుకంటే మనకు కావాల్సినటువంటి మినరల్స్ కానీ విటమిన్స్ కానీ అన్నీ కూడా ఫుడ్ నుండే వస్తున్నప్పుడు ఫుడ్ ఆల్సో కమింగ్ ఫ్రమ్ ప్లాంట్స్. సో ఇలాంటి అవేర్నెస్ అనేది మనం పెంపొందించాల్సినటువంటి అవసరం ఉంది. సో ఎనీహౌ, థ్యాంక్ యూ ఫర్ కండక్టింగ్ దిస్ టైప్ ఆఫ్ program. Dr. Tirupati Garu, and welcome to all participants.
Okay, thank you very much, sir. So you are quite differ from our side because of your belongs to commerce and business administration. Even though you are given a wonderful message regarding of this second one-day national webinar. So thank you very much, sir. Really, your support and also your cool administration and also supportness, kind-heartedness, memorable for my entire life. Thank you, sir. Now I would like to invite Dr. P. Nitin Garu, Vice Principal of our college. And please unmute yourself, sir. Nice. Okay. So now I would like to invite today's organizing secretary, Dr. Rautu Radhakrishna Garu, Assistant Professor, Department of Botany, and Head Department of Microbiology. So please welcome and unmute yourself, sir, and give a brief message. Welcome, sir.
Thank you, Tirupati sir. Welcome to this webinar. It is with immense pleasure and enthusiasm that I welcome you all the one-day national virtual seminar on pharmacognostic studies of medicinal plants 2025. This seminar serves as a platform for academicians, researchers, students, and industry professionals to share knowledge and insights into the fascinating world of medicinal plants and their pharmacognostic potential. The study of medicinal plants has been integral to traditional medicine system and continues to hold promise in modern drug discovery and development. This seminar aims to foster dialogue and collaboration and innovative researchers in a critical field. I extended my heartfelt gratitude to our distinguished keynote speaker, Professor Dhale, PG Department of Botany, SVPSS, Dr. Gogre Science College, Dhale, Maharashtra, and also my heartfelt gratitude to our dynamic principal and also the webinar pattern of this webinar, Professor Kalaputla Ramakrishna Garu. He is the principal of both colleges, SRR Gas Autonomous Karimnagar College and the GDS Women Jagityal College, and also my respected heartfelt congratulations to Dr. Jayakishan Hoja sir. He is the principal of GDC Godavarikani and Mantini also, and also my heartfelt gratitude to our vice principals, Dr. Nitin sir, HOD of Physics, and Dr. Rajay sir, HOD of Commerce. And also my heartfelt thanks to our convener of this webinar, our HOD sir, Dr. Tirukovala Srinivas, Associate Professor, HOD of Botany and COE of Control of Examination of this SRR College. And also my heartfelt gratitude to our Chief Guest, Dr. Tirunari Yugandhar sir, Associate Professor of Botany, KDC, Art Autonomous College, Hanmakonda, and also my heartfelt gratitude to my brother and very active, dynamic person, this coordinating secretary of this webinar, Dr. PADALA Tirupati Garu, Assistant Professor of Botany and NSS Program Officer, and also my heartfelt gratitude to our co-conveners of this webinar, Dr. MD Tahir Hussain sir and Kalajyoti Madam Garu. And I also thank all the participants for their interest and support in making this seminar a success. Let us make the most of this opportunity to learn, discuss, and contribute to the collective growth of knowledge and teaching and fruitful experience. Thank you one and all.
Okay. Thank you very much, sir. So, the huge experience personality, my beloved brother, Dr. Radhakrishnan sir, has clearly stated that his vision and also supportness is ever inspirable to all the faculty members in the Department of Botany and also Department of Microbiology. Thank you very much, sir. Now, I would like to invite my beloved brother, my friend, my lovable brother, Dr. Muhammad Tahir Hussain, recently who completed his PhD from the reputed university, that is Usmania University in the Department of Botany. I would like to welcome and please unmute yourself and give a brief message of this national webinar.
Thank you, Dr. PADALA Tirupati Garu, my friend and a kind of resource person to me as well. And I welcome all of the participants, including the patron, guest, keynote speakers, and conveners, and organizing committee members who are there. I welcome you all for today's wonderful day. Today is the day to enlighten the participants regarding the what we call the medicinal importance of the plant. In fact, plants are always neglected. There is no plant without any importance. It is the man who has to know the importance of that. Generally, for a layperson, plants are grown for ornamental or aesthetic purpose. But in fact, the plants are known for their pharmaceutical importance as well. As earlier Rajas are told, the Ayurveda is purely depend on the pharmacognosy of the plants only. So I really appreciate Dr. Radhakrishna sir for selecting such a topic and for inviting Dr. Dhale, the keynote speaker, a very knowledgeable and person who is going to enlighten us, all the participants. I hope all the participants will enjoy this session and all the participants will get enlightened. Even we too have the research scholars, we have the PG students. So we also request a kind of feedback from all of them as well. Because your feedback will provide, it is a kind of encouragement for us to make the seminar much more knowledgeable, much more living in future concerns. So thank you, sir. Once again, I thank all the dignitaries who were involved in organizing this. Thank you, sir. Over to Dr. PADALA.
Oh, thank you, my dear brother. Earlier, Dr. Muhammad Tahir Hussain Bhai has worked as a faculty FAC principal of GDC Mantani. In his tenure, GDC Mantani has accredited, NAC accredited by with B grade, B+ grade. So that's why the spirit and commitment and concern and dedication we observed from his side. So we are very lucky and we are very happy with his association in the Department of Botany. Our botany department also rigorously and also the very accelerated manner to reach the subjects, not only in the academic side, but also in the research side also. Why? Because in our botany department has the PG students, they are very actively involved in teaching learning process. Our botany faculty members in the guidance of the head Department of Botany, Dr. Tirukovela Srinivas Garu, all the classes, academic side, without any interruption. Even though the practicals in the morning session definitely done in the laboratory by all the faculty members, Dr. Kandukuri Kala Jyoti Madam Garu, Asampalli Swaruparani Madam Garu, Dr. Muhammad Tajuddin Garu, and also Dr. Rautu Radhakrishna sir, and myself, we are involved in the teaching learning process. All the students are deputed. All the students are deputed with their academic side, and also they are utilizing the fruitful time in the library also in the afternoon session. So that is the commitment we are observed in the Department of Botany. With these words regarding of our beloved brother Dr. Muhammad Tahir Hussain. Now I would like to invite Dr. Kandukuri Kalajyoti Madam Garu, who is going to introduce the eminent personality and legendary personality and hero of this today's national webinar, Dr. Professor Datta Dhale Garu. Please welcome Kandukuri Kalajyoti Madam Garu. Please introduce about today's keynote speaker and unmute yourself.
Thank you, sir. Thank you giving this opportunity, sir. Dr. P. Tirupati Garu. Myself, Kalajyoti Kandukuri, Assistant Professor of Botany, SRR Government Arts and Science College, Autonomous, Karimnagar. Welcome all the participants. Today's national online webinar on Pharmacognostic Studies of Medicinal Plants 2025 from the Department of Botany, SRR Government Arts and Science College, Karimnagar. On this auspicious occasion, I feel prestigious to introduce Dr. Datta Dhale Garu, today's keynote speaker. Datta Dhale did MSc Botany 2001, and his PhD 2008 in botany from Dr. Baba Saheb Ambedkar, Marthwada University, Aurangabad, with pharmacognostic as a subject of specialization. Presently, he is working as a professor and research guide at Postgraduate Department of Botany, SSV Santas, LK Dr. P. Gorge Science College, Dhule. He has consulted for expertise in study of medicinal and aromatic plants, pharmacognosy and phytochemistry. He honored 21 different awards like Research Excellence Awards in 2023, Award World Science and University Rankings in 2021, Academic Excellence Award in 2020, Distinguished Faculty Award in 2019, Best NSS Program Officer in 2019, Distinguished Research Award in 2017, Innovative Science Technology Awarded in 2016, Young Scientist Awards in 2016, Best Research Paper Award, etc. He recorded 129 different publications on name. He has published 141 publications, in which 3 patents, 11 books, 105 research papers in national and international journals, along with 22 book chapters. He also presented more than 55 expert talks at various workshops, conferences, and seminars. He actively participated in more than 125 seminars and conferences, beside many training programs. He also keen interest in editing and associated with journals of training programs. He has keen interest in editing and associated with journals of guides, different 23 journals edited, 15 special volumes of different international journals. He has guided 34 candidates for research projects for university academic degree MSc Botany. He is PhD guide in botany, now pursuing 6 students pursuing their doctorate in various topics. Additionally, he is finished 2 projects from UG Government of India. One project from Rajiv Gandhi Science and Technology Commission is going on. As a result of his curricular activities, he is working as a team leader from many national and state NSS camps. He is a member of many prestigious academic and scientific bodies and society. He also contributes for Marathi daily newspaper and create interest in plants by delivering takes, especially for young generation. Thank you, sir. Thank you giving this opportunity.
Okay. Thank you, Kalajyoti Madam Garu. So now I would like to invite Professor Datha Dhale Garu, the eminent professor. Please welcome, sir. And I will give a sharing option to your presentation, sir. So please share your screen, sir. Please welcome and unmute yourself, sir. Enabled, sir. Please unmute, sir. Please unmute yourself. Yeah. Screen
Is it visible, sir?
Yes, sir. It is visible, sir. Please, you proceed.
Okay, sir. Thank you. One day national GDC, Godavari Khani, Manthani. Dr. Ugandhar sir, convener of this program, Dr. Srinivas sir, head of department, and Padala sir, and convener delegates and dignitaries. Students.
Sir, excuse me, sir. Your voice is not clearly audible. Sir, if possible, sir, if possible, please, you left from this, please you leave and rejoin, sir, please, if possible. Your voice is not clearly audible. Due to some technical problem, sir is going to join again. So please wait for 2 minutes. Okay, sir has joined. Sir, please unmute yourself, sir. And Janayar, sir, Janayar. Okay, sir, please unmute yourself, sir. సార్ దగ్గర నెట్వర్క్ ఇష్యూ ఉన్నట్టుంది. Okay, sir. Your screen is visible. Please click on slideshow. Sir, your voice is not audible. Dhale, sir. Sir, please unmute yourself, sir. Okay.
Yes, sir. Yes, sir.
Yeah, you are clearly audible, sir. Please, you share your screen, sir.
Thank you, sir. I think there is a network. I am going to present my presentation. That is the pharmacognostic studies on medicinal plants 2025. As the principal sir told, study of structural, physical, chemical, biochemical, and sensory character of natural drug. That is in the crude form, plant and animal origin. That is distribution, cultivation, collection, identification, preparation, evaluation, preservation, and use and commerce of the medicinal plants. In this way, pharmacognostic work on the crude converted into the drug and pharmacognosy Pharmacognostic.
Sir, your voice is not audible, sir.
Let us start.
Sir, your screen is not visible, sir. Please share your screen, sir. Okay, sir. Proceed, sir.
Okay, sir. Written divided into Western medicine, the Unani medicine, the Ayurvedic medicine, the Orient medicine, and the Greek and African system of. Let us. We are. Point that is known as the evaluation of drug. Evaluation means the substance becomes as a drug if it changes the pathological or the disease state of human or animal to the determination of its quality, purity, and detection of nature of adulterants by various parameters like morphological, microscopical, and biological. In this, in the pharmacognosy, we are going to concentrate on the medicinal plant. The medicinal plants is any plants in which one or more of its organ contains substance that organization which present in the word can be utilized as a medicine if it is used an accurate quantity, that is a dose, or used with other proper combinations and in purified. And 50% of the tropical plants having 250,000 plant species, at least 10,000 means there is a vast scope are endemic plants, 7,263 total medicinal plants. About 2,000 medicinally important plants have been used. 70 plants market. There out of these 2,000 medicinal plants, needed daily in market, and traditionally in different therapies, that is tradition therapy known as the Rigveda, having used 67 medicinal plants. Charak Samhita 1000 Ayurveda about 1200 to 18 way of functional pharmaceuticals. We found that 270 species utilized by different pharmaceutical industries, and out of these have one twenty
So please wait for 1 minute. Due to net issue, Sarah has left the meeting. Sarah is going to join within 2 minutes. జాయిన్ కాలేరు కదా. Okay, due to net interruption, SAR is waiting to join. So in this webinar, so many our eminent personalities of our Department of Botany of various colleges. At the same time, other than Botany department also, I observed in this webinar. Sarjanayada. Okay. Sir, please, Dhale sir, please unmute yourself, sir.
Yes, sir. Yes.
Okay, sir. Please share your screen.
Yes, sir. Today is here big issue of network. How I? Okay, sir.
Yes, sir. Please, you may.
Yes. Okay, sir. Thank you, sir. This is cumulative data, which is depends on the on survey of market, and I am showing here the top 10 trading medicinal plants. Why I am going to show here top 10 medicinal plants? There is a huge demand for this plant, and with the help of pharmacognostic, we can find alternative to this plant. This consists of the Embellia ribens, Cassia sena, Asparagus racemosus, Terminalia tubula, Withania somnifera, Glycyrrhiza glabra, Gymnema sylvestris, Azadiracta indica, Tribulus terrestris, and Vetiver gingiunide. These are the top 10 plants of the Indian having the huge demand, and it is necessary to find out the alternative of these plants with the help of pharmacognostic. Then nationally important plants found in Maharashtra, these are the Agelmarmensis, Andrographis paniculata, Asparagus racemosus, Bacoma moneri, then Chlorophyton borevillanum, Coleus barbatus, then the Comifera embleca, then the Embellia, then the Emblica, then the Gloriosa. These are the some nationally important medicinal plants found in Maharashtra. We are going towards the some misunderstandings which are found in the market. That is the well-known misunderstanding about the Bacopa moneri and Centella asiatica. Everyone knows that the Brahmi is an important plant. In various advertise and some many of the nursery vans showing you the Bacopa moneri, which is a real Brahmi, instead of these giving us the Centella asiatica. Centella asiatica has have nothing a phytochemical which is known as a Brahmin. Brahmin is only present in Bacopa moneri, which is found in first photograph. But the everyone of in the market which provide Brahmi in the form of this Centella asiatica. Then Asparagus racemosus, which is known as Shatavari, is a reputed medicinal plant which has having a steroid containing drug. Then the Gudmar, Gymnema, most of the peoples didn't trust on the Ayurveda or didn't trust on the medicinal plants because they're showing the very late result. For those, this plant is a very important example which known as common name Gudmar. Gud means sweetness, Mar means killing. The plant which having the capacity to destroy the sweetness within a minute. So, as a student of botany, as a student of life sciences, you may know go towards this plant because it having a very particular and definitely character which lowers your high blood sugar level within 1 minute and destroying all the sweetness from your mouth within 1 minute. This plant may also play a beneficial role in diabetes. Then another endangered species, which is known as Sarpagandha, Rauwolfia serpentina. It having a vast number of the chemicals found in these roots of the plant, and these roots or the plant chemicals, phytochemicals found in this plant have the amazing result on the brain for the controlling the brain power and different properties this having. So that is necessary to propagate this plant, which is a very important plant. Then Vitex nirgudi, which is a very well-known painkiller. While getting pain, we are going towards the medicine and taking a tablet. Instead of that, you must go towards the Vitex nigundo, which is a very well-known painkiller. Then there is the same confusion in the Ashoka. The second picture showing the Pongamia Polyanthus longifolia, which is an garden or ornamental plant. Instead of this, the drug tree, which is known as the Saraka Ashoka, which is showing in the first picture, which having the very well-known properties. The famous Ayurvedic drug, which is known as Ashoka Arista, which is prepared from this Ashoka, Saraka Ashoka, bark of this plant. And today there is a vast demand or the very large amount of demand of this plant. So it is necessary to study the alternative for this Saraka Ashoka because the drug is in its bark. Some of the plants having same plant with different names. When we are going to use the bark of this plant, which is known as drug name Kudasal. The plant is same plant, which is known as Holarina antidysentrica or Holarina pubescens. Whenever we going to use its seeds, its name is an Indrazava. The Kudasal have the different property and the Indrazava have the different properties. So one plant have different properties in its different organ, and this is knownly with the help of pharmacognosy. So it is necessary to understand the importance of pharmacognosy with the help of these different importance. The well-known antiviral plant, which is known as the Phyllanthus amaranthus, then we must go towards the natural colors instead of the synthetic color with the help of this Pixa orelina, which gives us and the sindur or red color, which is edible color. Then Butea monosperma. In the many of the cosmetics, you must use the petals of this palas, which is known as Butea monosperma. And these are the some reports from the ancient medicine or the traditional peoples, which shows the Butea is must used in the cosmetics also. You can also go towards the some home preparations like the Emlica, Amla candy, and so many. Then some of the tribal peoples preparing their own medicine with the help of medicinal plants. This is the well-known medicinal plant which is known as Jyotishmati, Silastrus paniculata. This is the tribal people which prepare the oil from these seeds of this Silastrus paniculata, which is used for the different alignment. The common name of the Jyotishamati indicates Jyotish means future and Mati means brain or Mati means Buddhi. It means the plant which is useful for the enhancing the power of brain is used, and these peoples are used this oil for different alignments. They prepared oil with the help of this instrument which is in the hand of this common man, and the churna, which is known as the powder, is prepared from these seeds, and putting this powder in this instrument, they mechanically pressed and oil is extracted from this seeds, which is used for so many preparations. There are so many plants reported by the Ayurvedic Drug Manufacturing Association, giving the list of 255 medicinal plants which have the daily need in the market. So I am showing here just list to work on these plant and also to work the alternative of this plant, because for the continuous demand of this plant, there is a vast need to identify this plant properly and give the alternatives. And for this proper identification and proper content, I written a monograph which consists of 255 plants which are used for the Ayurvedic Drug Manufacturing Association, and the book is consist of the trade names to scientific detail. It means ADMA gives you the drugs with the help of the common name like these, Brahmi, then the Babulsal, the Dhale, but they didn't showing there any scientific detail of this drug. So that's why I am giving to a literature for the correct identification of these plants. There is a vast confusion in the market or in the academics also. There are some of the professors also working on the plant, but they didn't know which plant they have taken for the research and which they are actually working. Some the paper I am showing here is also present in internet also today. He's showing the study of Anacalus pyrethrum, but whenever he going to study Anacalus pyrethrum, he showing the pictures of different plant, and this is not Anacalus pyrethrum. Anacalus pyrethrum is this one. So whenever we are going to study on the plant, it is necessary must identify with a proper taxonomist, and most of we depends upon the Google search and the lens search. I am searching about for the plant which is known as the Mallotus philippines, which is showing in the corner, and the Google shows me the result. Previously I told you the plant is known as the Bixa, and Google showing here for Mallotus philippines. In this way, we doesn't depend on the Google. In this way, there are so many examples which I found on the Google. They are misly identified. And not only the Google, there are some workers. They are also didn't properly identified, but working and publishing the articles on different journals. And also there is a vast mix-up of vernaculars name. There are so many examples which found the similar vernacular name, but the plant have the different. Here I am showing so many examples. Just I told previously Dacopa moneri, which is known as the Brahmi, which alternatively mixed with the Centella asiatica, and there are so many examples like the Shankapushpi. There are so many plants which is commonly known as Shankapushpi and birch. There are so many examples which having mix up of the vernacular name. And for this, all this confusion have been resolved with the help of pharmacognosy. So pharmacognosy have studied proper identification, determination of its quality, determination of its purity. So for proper identification, you must go towards the Botanical Survey of India, then PG department, where and a taxonomist must have a botanist, which is having a specialization taxonomist. So for the proper identification, you must go towards the botany department, which is an taxonomist, or you must go towards the Botanical Survey of India. And for going towards the identification, you must prepare the herbariums, and herbarium have some norms with the help of, here I am showing the herbarium sheet and it having some of the particular norms. While going towards the identification, you must prepare an herbarium, and herbarium must be pressed in suitable way. Then there are some names which is recently changed. For example, Acacia catechu, now the name is changed to Senegalia catechu. Then there are so many examples. The Acacia is genus is converted or the changed as the Senegalia. There are so many examples. The Acacia nilotica genus is changed Vasilia. Then the Terminalia arjuna is the Terminalia cunentia. So many examples. You must go towards these names which having recently changed. While we are going towards the study, we use traditional names. So the pharmacognosist must knowing the proper identification and proper nomenclature. Then the pharmacognosist must know the quality. The word quality refers to intrinsic value of the drug. That is the amount of medicinal principle or the active constituents present in the drug. And these principles are classified as the proteins, carbohydrates, alkaloids, glycosides, tannins, amino acids, saponins, phenol, flavonoids, volatile oil, lipid, antibiotics, steroids, and so on. And with these phytochemicals, you must go towards the quality control with the help of different phytochemical tests. And for this, it is necessary to identify its purity. Purity of drug will be achieved, can be achieved by proper identification and quality assurance. For the evaluation method, you can go towards the different analytical techniques. That is, first one is the organoleptic, second is a microscopic, third one is a biological, fourth one is a chemical, and fifth one is a physical. For the organoleptic evaluation, you must go towards your 5 sense organs. And these 5 sense organs gives you a proper identification. That is the identification of shape, identification of size, identification of color, identification of factors, and internal color, odor, and taste. In this way, with the help of this organoleptic character, we have to identify bark or stem, underground structure, leaves, flower, fruit, and seed, or its different structures. You can identify it with the help of these 5 sense organs. And for the microscopic evaluation, you can go towards the detailed microscopic study with the help of different transfer sections, different dermal studies, and microscopic or microchemistry. In this way, you can also, in the microscopic evaluation, you also study powder plus reagent or powder plus chemical. In the histochemical detection, you can observe the starch grain, aleurone grains, fats, fatty oil, volatile oil, and resins, calcium oxalate, then lignified cell walls, then cellulose cell wall, mucilage, and tannins. All these characters have been identified with the help of histochemical evaluation. While in the dermal study, you can identify the leaf epidermis, or dermal study, you can identify the palisadal ratio. With the help of palisadal ratio, you can give and data which is useful for the identification of proper plant. Then vein islet number, vein termination number, stomatal number, stomatal index, and the Lycopodium spore method, which is an biological spore method. In this way, in the epidermal tissue system, you can study the epidermis or epidermal with the help of stomata, trichome. Trichome may be glandular or non-glandular. Particular trichomes are found in particular plant species. Particular stomata are found in particular genus and genus species, it means they are properly used for the identification and authentication of genus and species. And for this, stomata having, there are different types of the stomata. I am showing here only few types of the stomata, and this few type, with the help of these few types of stomata, you can go towards the authentication of a particular plant. You can also identify the palisadal ratio. How the palisadal ratio is calculated? Palisadal ratio is just calculated with the how much palisadal cells connected with the epidermal cell. Here we can see in the example, here 18 palisadal cells have beneath with the 4 epidermal cells. It means it having the palisadal ratio 4.5. In this way, you can fix this palisadal ratio for a particular genus or particular species. In this way, you can observe maceration of the stem. With the help of maceration, you can observe the different types of the vessels, and these vessels have the remarkable identification characters, and the particular plants have the particular vessels type. So with the help of pitting on the vessels, you can definitely identify the plant which have been studied or which have been standardized. Here, with the help of microscopic material, you can identify these vessels, and with the help of the vessels, you can going towards the standardizing the plant. For the standardization, there is an anatomical characters. For the anatomical characters, you can go towards the root anatomy, stem anatomy, node anatomy, leaf anatomy, and petiole anatomy. For a particular plant, the anatomy of particular part is an fix. So whenever you are getting the adulterated plant, you can definitely, with the help of simple transfer section, you can identify the adulteration. It means the anatomy gives a very best tool for the standardization of drug in pharmacognosy. Then the third one method is an chemical evaluation. Chemicals have the crucial role in the plants, and they also have the crucial role in identification. So this techniques is very useful for the standardization of the drug. Then the standardization of the drug or the phytochemical extraction will be analyzed or calculated with the help of extraction, isolation, and the structural elucidation of natural product. That is with the help of MS, mass spectroscopy, NMR, then or their various chromatography like the TLC, HPLC, then the LCMS, and so on. Then extraction is the separation of the medicinally active portion of the plant or the animal tissue using the selective solvent through the standard procedure. And this gives the separation of phytochemicals for the identification or drug discovery. And for this, standardization or standardized extraction is done. The purpose of standardized extraction procedure for the crude drug, that is the medicinal and aromatic plant, to attain the therapeutically desired portion. It means in the crude form, it having the large amount of mass. With the help of standardization, you can limit its mass constraint. To eliminate unwanted material by the treatment with a selective solvent known as maelstrom, the extract thus obtained after standardization may be used as an medicinal agent. And this general techniques of the medicinal plant extraction, you can take one or the many of the procedures. For example, maceration, infusion, percolation, digestion, decoction, hot continuous extraction, aqueous alcohol extraction by fermentation, then the counter current extraction, microwave assisted extraction, ultrasonic extraction, supercritical fluid extraction, then the phytonic extraction. In this way, you can extract the medicinal plants with these different methods. And the steps involved in the extraction of the medicinal plants have size reduction, extraction, filtration, concentration, and drying. And due to this, the huge amount mass which is going to convert into the mass which is able to put into the capsule with the help of these techniques. And some of the parameters for selecting appropriate extraction method, that is the authentication of plant material by botanist, then use the right plant part, also the age of the plant, because the active drug is only active within a certain limit of the plant. When a plant having annual, or plant having an herb, or plant having shrub, it is necessary to harvest plant at a particular time, particular season, and also necessary to collect the plants from particular place also. The nature of its chemical constituents, then the grinding method and powdering technique, nature of constituents, that is the polar and non-polar solvents may be used, the quality of water, that is the menstruum, the design and the material of fabrications of the extractor, then the analytical parameters of the final extract with the help of different techniques. Then you can also use the chromatography for herbal drugs. It may be used for the separation, identification, impurity detection, and assay of herbal drugs in the formulation or in the extract are carried with the some instruments which are present in the different labs, which are useful for the identification of phytochemical constituents which found in the different drugs. Then the qualitative chemical examination have been determined or examined with different tests. For example, detection of alkaloid, detection of carbohydrates and glycoside, detection of phytosteroid, detection of fixed oil and fats, detection of saponin, detection of phenolic compound and tannins, detection of protein and free amino acid, detection of gum and mucilage and volatile oils too. Then the next important part of the pharmacognostic study is an biological evaluation. Whenever you have identified the proper plant, whenever you have standardized a plant, then the particular chemical which is found in the plant it is necessary to evaluate its biological character, because whenever you are not going towards the standardization of the biological parameters, you cannot discover a drug to formulate in a market drug. So it is necessary to evaluate a drug for its biological efficiency. The biological efficiency of drug is very useful in determination of pharmacological activity. Since living organism or their isolated living tissue are used, this method is called biological method, or also known as the bioassay. Many drugs, particularly the antibiotics, toxins or toxides, also vitamins, are assayed by this method. And this bioassay is, as I told, you can evaluate this bioassay with the different techniques. That is the one of the technique is antimicrobial activity. Antimicrobial activity refers to process of killing or inhibiting the disease-causing microorganism. And with the help of various antimicrobial agents are used for this purpose. And antimicrobial activity may be in antibacterial, antifungal, or antiviral. They have different methods of action by which they act to suppress the infection. And you can This test is a very easy test to do in our undergraduate students also and postgraduate students also. Then the antioxidant. In the same way, the biological assay have been identified with the help of antioxidant activity. Then our fifth one evaluation method is known as the physical evaluation method. In the physical evaluation, it is a necessary or it is a very useful to standardize the drug, or it is a very useful to find out the adulterants from the drugs. The physical evaluation of approved drug is accomplished by the determination of various physical characters using various physicochemical techniques. For example, specific gravity for fats and volatile oils, melting points for the alkaloids, optical rotation of alkaloids and volatile oil, ash value, and the moisture content. There are so many physical parameters like the viscosity, melting point, solubility, moisture content, and volatile matter, specific gravity, density, optical rotation, refractive index, particle size determination, bulk density, bitterness value, homolytic activity, swelling index, homing index, ash value, astringency, organoleptic testing, determination of ash value, pH estimation, estimation of foreign organic matter. In this way, in the physical evaluation, there are lots of the methods by which you can go towards the standardized and drug. Here we can see some of the examples in which we can go towards the standardize the drug with the help of physiological or physical test, in which the viscosity of a liquid is constant at a given temperature and is an index of its composition. Hence, it can be used as a means of standardizing liquid drug. It means viscosity of the honey is different from the viscosity of oil. In this way, you can understand the technique which is useful for the standardization as an viscosity. The melting point, you know that the melting point of different drug or different material have its own melting temperature, and with the help of this own melting temperature, the standardization value gives you an very well-known standardization instrument or parameter, which is an easily calculated in your laboratory with an very simple instruments. Then the moisture content and volatile oil. Moisture content means drug consists of iron water particles. For this moisture content, it is determined by the heating drugs 105 degrees Celsius in an oven for a constant weight. And in this way, this gives a standard quantity for the pharmacognostic standardization. Then the ash value. The ash value gives you the purity of the drug or the minerals, purity of the drug, in which the amount of the minerals present in the drug. And this gives you the detection of low-graded product, then adjusted product, excess of sand, then the earthy matter, and these matters can easily evaluated with the help of this ash value. Then how the ash is prepared? Just put in muffle furnace at 600 to 650 degrees Celsius for 3 to 4 hours, and it gives you an ash. And the ash is also determined for the different parameters. That is the acid insoluble ash, acid soluble ash, and there are so many different methods which are giving to evaluate the different parameters of the ash, and which gives and standardization or the standard parameters with a particular reagent. Then in this way, you can also evaluate the water soluble ash. Then the determination of extractable matter. How much drug consists of extractable matter? It means if a drug consists of extractable matter in large amount, it gives high amount of drug. It means it is necessary to produce large amount of the drug from a few amount of the crude drug. And this can be done with the help of different techniques, that is the cold method, hot method, and Soxhlet method. The cold method is just putting the material in the flask for the 6 hour or up to the 24 hour or 18 hour. Different methods given for the different herbal drugs. It means some of the drugs have capacity to dissolve within few hours, or some of the drugs having capacity to take more and more time to dissolve in a particular solvent or in a particular extract. In this way, this is a very simplest method, which is known as the cold maceration. Another one is an alcohol soluble extract tube. 10 gram of air dried drug, coarsely powdered, was soaked with 100 ml of alcohol in conical flask for 24 hours. It gives meanwhile it gives shaking, it gives an extract which is alcohol soluble extract. And this most of the compounds like the alkaloids and the different lipids, fat, which are not able to easily solve or easily soluble in the water or the other solvent which are easily soluble in alcohol. So it is a very suitable technique or a very valuable technique which is known as the alcohol soluble extract tube. Insoluble matter you can also calculate from soluble to insoluble. And last one is the total solid content. How much solid matter present, the residue obtained when the prescribed amount of the preparation is dried to constant weight under the specific condition gives you and total solid content. Then water content. How much amount of the water content in which present in an drug or crude drug, and it gives a particular drug have the specific capacity to absorb the moisture or the water. So it is an best tool to standardize an herbal drug. Then hot extraction. In the hot extraction, whenever cold extraction not working, then the solvent extraction not working, then we can go towards the hot extraction method, because those phytochemicals which are present in the very less amount in particular drug, they can be extracted with the help of hot extraction method, or another one is an hot continuous extraction, that is known as the Soxhlet extraction. It means in the hot extraction and in hot continuous extraction, there is a little difference. In hot continuous extraction, there may be cycle continuously run up to the 4 to 84 hours, up to many of the cycles. And that's why whenever drug contains a very small amount or main active ingredient of the drug is present in the very small amount, in this method you can get the very less amount drug can easily extracted in this hot continuous extraction, which is known as Soxhlet extraction. Then another is an infusion. It is the just putting the preparation, which is known as maceration, that is the infusion. Then decoction, everyone known as the what is the decoction. We prepare every day. Tea is nothing but the decoction. In the same way, you can use this method for preparation of drug extract, which is known as extraction. Then the swelling index, a particular plant or the particular material or particular part of the plant have its definite swelling index. And this swelling index is an also important parameter for standardizing the drug. Then water and volatile matter. Volatile matter which is present in the aromatic plant is evaluated or is analyzed with the help of Clevenger apparatus. Then every plant with the help of active ingredient, while the picker or the tribals, whenever getting the active ingredient, they may getting some of the undesirable parts, and these undesirable parts is known as foreign organic matter. And this is may be due to improper handling of the drug, or it may be, and it is necessary to calculate the organic matter, foreign organic matter, which is necessary for our pharmacognostic evaluation. Then for the evaluation of an herbal drug, these are the 5 techniques. First one consists of the organoleptic. In the organoleptic, you can analyze the color, odor, taste, texture, facture with the help of our different 5 sense organ. In the second one, in the botanical, you can go towards the microscopic and macroscopic analysis. In the macroscopic analysis, you can note down the shape, external and markings of the plants. While in the microscope, you can go towards the qualitative, quantitative, also for the same, that is scanning electron microscope study and powder study. While in the physical parameter, you can go towards the moisture content, extractive value, ash value, then the fluorescence analysis, etc. You can done. There are so many parameters which you have seen just in physical study. So physical parameters are very standard parameters, are easily getting the results for the standardization of drug. Then the chemical. In the chemical, you can go towards the qualitative, quantitative, or the chromatography. And in biological, you can go towards the analgesic study in bacteria, fungi, or viruses, or microbial contaminations, then the pharmacological evaluation, then the toxicological evaluation. In this way, you can, in the pharmacognostic, or for the pharmacognosy means the standardization of drug, herbal drugs, or standardization of herbal drugs, you can study the drug with this 5 aspects. And considering these 5 aspects, you can easily study it on different medicinal plants. And for this, I am studying some of the references for you published, also going whatever I do research frequently published on the radios and different channels, and whatever I doing research around my state, I frequently publish it in local newspaper, by which we can understood the common peoples what around us, so they can aware about the botany, they are aware about the plants. Also we have done the some of the plants which picks the fruits, the seeds, and germinated in laboratory or in the garden of our department, and they gives a very beautiful result. And this result shows here the endemic or endangered plants which are found in our forest also conserved in the garden also. And doing this study, it is necessary to publish your work in different ways. Just the our principal sir, the Ramakrishna sir, says they have observed or they have studied my CV on net. With the help of, they can understand me. In this way, the new researchers or the students and the workers who are going towards in their field must publish their work in different or standard journals, local newspapers, and whenever you get the chance. In this way, you have to publish your work with various platforms. And lastly, I just told, eat your food as your medicine. Otherwise, you have to eat medicine as your food. As the here students having the different disciplines, the message is very important. So with this message, I would like to stop my presentation. If you have any query or you have any question, you may ask. I am with you. Thank you, sir. Can I stop my sharing?
Sure, sir. Thank you very much, sir.
Okay, sir.
Regarding of today's excellent national webinar, if you have any queries of from all the delegates, please you may feel free and ask or discuss with our eminent professor Dhale garu. So I will give an opportunity to unmute yourself to all the dignitaries. I given a chance to unmute yourself and interact with our respected keynote speaker. Okay, I have given.
Sir, Dr.PADALA, sir.
Yes, sir.
Congratulations, sir. You are very, very informative and very excellent, sir. And you have lot of slides are very, very interesting, sir. Thank you very much, sir. On the behalf of our botany department and our college, we are always thankful to you, sir.
Thank you, sir. I am very sorry to due to network issue. Firstly, I got very difficult to connect, continuously connect. So I am extremely sorry for inconvenience.
No, sir. The technical is in not our hands. Anyhow, very, very thankful to sir.
Yes, sir.
You are very informative and very excellent slides also. Okay, sir. It definitely help to our participants.
Yes, sir. Thank you, sir.
Thank you, sir. Anybody else? Okay. So now I would like to invite Dr. Asupali Swaruparam to propose a vote of thanks to all the delegates and the organizer committee and patrons of the college. Please welcome madam. Vajita madam, are you ready? Any point from your side? Okay. So, madam,
thank you, sir. Dr. P. Tirupati, sir, for giving this opportunity. Good afternoon, everyone. Myself, A. Swaruparani, lecturer in botany, SRR Government Arts and Science College, Karimnagar, Autonomous. I express my gratitude to all the dignitaries and the participants on today one-day virtual national webinar on pharmacognostic studies of medicinal plants. I thank to Professor K. Ramakrishna Garu, Principal of SRR Government Arts and Science College, Karimnagar, Autonomous, and Professor K. J. Kisan Oja Garu, Principal of GDC, Mantini. My heartfelt gratitude to today's keynote speaker, Prof. Datha Dhale Garu, who has given valuable information on pharmacognostic studies of medicinal plants. He enlightened us with numerous plants like Brahmi, Saraka indica, and Asparagus, Azadirachta indica, and herbal techniques. I'm also thankful to today's webinar chief guest, Dr. T. Yugandhar Garu, I thank to Vice Principal Dr. T. Rajay Garu, SRR Government Arts and Science College, Karimnagar. And thank to Dr. P. Nitin Garu, Vice Principal of SRR Government Arts and Science College, Karimnagar, Autonomous. I am also thankful to convener of today National One-Day Virtual Webinar, Dr. T. Srinivas Garu, HOD Department of Botany, Crop Production and Food Science, and Controller of Examination, SRR Government Arts and Science College, Karimnagar. Thank you, sir, conducting such an informative webinar on pharmacognostic studies of medicinal plants. I'm also thankful to co-conveners Dr. Mohammad Tahir Hussain, sir, and Dr. K. Kalajoti Madam Garu. I'm also thankful to Organizing Secretary Dr. Raut Radhakrishna Garu. I'm also thankful to all the organizing committee members. Thank you, one and all.
Okay, Swarupa Madam Garu, thank you very much. So all the dignitaries, thank you very much for your unwavering support and cooperation and coordination of successful second national webinar. Little bit of net issue, even though our eminent professor Dr.PADALA sir has explained very excellent, and also the slides are very, very bigger, and each and every slide is very effective and also informative slides. We are utilize this wonderful opportunity. This time is very fruitful. We hope as a organizing secretary of this national webinar, and all the patrons, and even though all the delegates, they are satisfied and full. We are very happy to fulfill our actual aim. What type of the phytochemical compounds which are extracted from the different plant pods, and from the ancient India to the modern India, Professor Dr.PADALA sir has clearly elaborated very excellent manner. So thank you very much, sir, for your valuable time, your sharing your views, and also your huge experience regarding of this national webinar. We are fruitful. Our aims and objectives are fruitful. We are enlightened. All the dignitaries are very happy. So within 1 minute, we're going to share feedback link. So all the participants, we are requesting you, please fill the feedback link. It is very useful to, we can modify or we can change, we can add any other flavors to our upcoming national webinars, and even to the other programs from our department, from SRR Government Arts and Science College, along with the collaborative college, that is GDC Godavarikani and Mantani. So thank you very much. So I will share the feedback link. So Professor Dr.PADALA sir, we are very thankful to you, sir. Our patrons and convener, Dr. Tirukovela Srinivas sir, has chosen a wonderful, eminent personality from Maharashtra, from Dhale. So we are very happy and enhanced new knowledge regarding of this one-day national webinar. Thank you very much, sir. So I will share the feedback link. Please wait for 1 minute. Yeah, just now I shared the feedback link. Please click on this feedback link and give your feedback in the feedback link. So it is very useful for our programs, and also you may get e-certificate through your registered mail. And one more request from our side. some of the participants you may not get today because every day only 100 certificates are going to serve each day. So that's why more than 491 participants, they are registered this national webinar. So bottom of my heart, I am very thankful to each and every registered participant and the successful of this national webinar. You may get e-certificate, even though is there any mistakes occurred in the certificate, we can going to rectify. Maybe our aims and objectives are definitely feel fruitful. Thank you very much for your cooperation. And also all the participants from the Mantani, GDC Mantani, GDC Godavarikani, and our SRR Government Arts and Science College, Karimnagar, all the departments, teaching and non-teaching staff, and supporting faculty members, technicians, and conveners and co-conveners of this national webinar, and patrons, and chief guest, Dr. Tirunahari Yugandhar, Associate Professor from Kakatiya Government College, Hanumakonda. So each and everyone, thank you very much for these few words from my side. Thank you very much. So please, those who are fill the feedback link, you may leave with pleasure. Thank you very much. We expected from your side your support and encouragement as usual. Thank you. I hope you are all filled the feedback link. So with your permission, I will going to wind up the session. Maipal Reddy sir. Revati Madam Garu. Subhashini Madam Garu. Regular Sunita Madam Garu. Kethireddy Narendra Reddy sir. Dhale Vani Madam Garu.
Shall we leave him?
Subhashini Madam Garu.
Sir.
Is there any queries or you are fill the feedback link, madam?
Yes, sir.
Thank you. Thank you for your cooperation and support, madam, for grand
Thank you,
success of this second national webinar.
Okay, sir. Thank you.
Swaruparani Madam Garu. Meerenu. ఇంకెవరు తెలుసు? పద్మ, అభిరామి, Juveria Fatima Madam, Bhagyashree Madam, Halima Madam, Apurva Madam, Revati Madam. Okay, I hope you are fill the feedback link. Please respond. Otherwise, I will going to wind up the session. Rahul Devender sir. Dr. Rahul Devender Garu. Dr. Bunga Tirupati Garu. Dr. R. Sunita Madam Garu. Rekha Madam Ji. Munigal Archana Madam Garu.
Sir, once again send it in WhatsApp.
Is there any problem, madam? Okay. So thank you. With your permission, I will go into wind up the session. Thank you.